So when we think about parasites and infectious diseases, it's not hard to imagine the impact that parasites and pathogens have had on human history and human health. So they've been a part of our history for a long time, and they have really significant effects on global health. Both micro and macro parasites, for example, the schistosomiasis, infects hundreds of people globally. But plants are not immune either. We have, it's so weird, it's on a timer. Anyway, so the plants, plants are affected by a variety of pathogens, some of which have significant effects on crop yield and economic losses. Obviously, the veterinary industry is concerned with parasites, some of which are mere nuisances, but there are other effects that are more dramatic. And then, of course, wildlife populations are also affected by parasites and pathogens. So this is a famous quote by Price saying that most species on our planet are probably parasites. So in thinking about the impact that parasites have on a host ecology and evolution, you can also then study the effects that hosts have on parasite fitness, transmission, and their ecology and evolution. So one classic example that Clement will tell you more about probably is the effects that parasites have on host morphology and behavior in ways that potentially increase transmission. Parasites, like predators, have been shown to regulate the host populations. So there's some evidence of parasite host regulation. And then on an evolutionary scale, we see evidence for evolutionary arms race between parasites and their hosts in these dynamics called red queen dynamics. On an ecological level, I need to turn off, I don't know why it's doing that. So the ecology of the host can also affect parasite transmission. One hypothesis called the dilution hypothesis posits that the diversity of the host community can influence the transmission of certain pathogens, as in the case of Lyme disease. In turn, parasite host ecology can influence parasite evolution. So these monarch butterflies are infected with a protozoan parasite that has very high pathology. So not surprisingly, there's a culling effect during the migration process where many of the ones that do survive are inherently the resistant ones. So you end up with host migration behavior patterns influencing the evolution of virulence. The other aspect of, the other way to study parasites, which not many people have considered, but is now sort of an emerging field, is to bring together the field of physiological ecology, or ecological physiology, and disease ecology. And some of the questions that emerge from these include the obvious, which is what are the physiological effects that parasites have on host populations and host bioenergetics. So one disclaimer I want to put forward is that I am not a physiologist, and I managed to get a bachelor's degree without taking any physiology. So keep that in mind when you ask me certain questions. So I'm interested in what are the ecological and evolutionary consequences of resource allocation as it relates to host bioenergetics, and how do parasites affect that allocation of resources. So you can imagine that there's a certain amount of resources available to a host, and some of it is spent on somatic maintenance and growth. Some of it is just lost to heat, and then some of it is allocated to reproduction. But inevitably, some resources are directed toward defense, and that could be immunological defense, behavioral defense, and all of that ultimately determines an organism's fitness. So when resources are being shunted toward defense, obviously there's less of the pie that can be allocated to other aspects of the life history. Other questions that the students in my lab really have taken interest in is whether or not parasites can influence their hosts simply by living among parasites. So even in the absence of infection, can we measure certain indirect costs associated with infection? And then the bigger sort of question is, can we use metabolic theory of ecology, which is a theory or a hypothesis where host metabolic rate or metabolism and metabolic rate of an organism can potentially predict some of the ecological and evolutionary ecological processes and patterns in the natural populations. So the question is, can we apply some of the fundamental questions and predictions to parasite ecology? So I won't talk too much about this today, but more so about the first three questions. Of course, I should acknowledge the people who do the real work in my lab. This work has really been driven by Colin Horn and Taylor Brophy, as well as this exceptional undergraduate student, Monika Merciuski. I practiced that. As well as the support of other lab members. This work would not obviously have happened without their contribution. So the study system, as Mike mentioned, involves cactus, specifically the Saguaro Cactus. These are the big ones in the southwest desert with the arms. This one's probably 50 to 80 years old. They don't form their first arms until they're about 50 years old. So they can grow, quite tall. Now, sometimes they develop injuries, or in the case of Phoenix, Arizona, people use them as target practice. So sometimes you see bullet shells on the ground. And so they, when they sustain injury, they rot, just like any plant would. And that rot attracts all sorts of microbes, as well as these mites, Macrocheles sabatius. And the mites come with the flies, which is Drosophila nigrosporacula. The flies use the plant for mating and ovipositioning. When they arrive with their mites, the mites get off and they reproduce in the media until the media deteriorates to the point where the mites have to get the heck out of Dodge, in which case they get a free ride from the flies. So here you can see there are 2 mites attached to this fly. And as the rot ages, as the conditions deteriorate, the flies are much more motivated to leave with a fly. And under those conditions, the prevalence and the intensity of infection can increase dramatically. So these habitats, as you can imagine, are highly ephemeral, and the parasites utilize that resource until they can't anymore, and then they become parasitic. So when they're in the cactus media, they can reproduce, and they live as predators. They feed on nematodes that the flies conveniently bring with them too. So, and they're not specific to this species of flies. You can find Macrocheles sabatius in a number of insects. But it is a natural association that you find in the southwest deserts. So as I said, they use the host for dispersal. The mites can't get very far on their own, and while they're on the fly, they get a nice little meal out of it. And as the rot advances, so does the intensity and the prevalence of infection in the host population. So the female mites are the primary infective stages, and when flies are infected, they suffer a significant fitness loss. The males have reduced copulatory success, so that's not hard to imagine. It's like a really bad case of crabs. And they do just physically have trouble mounting females. And the duration and intensity of infection directly affects the severity of the pathology. Okay, the primary line of defense that flies have against these ectoparasites is behavioral resistance. So when they encounter, this probably won't work. It's meant to be a video, but it never works on PC. Oh, maybe it will work. No, it's teasing me with an arrow. So this is just some mites sort of cruising for flies, and they're running around, and this one has a few mites on it already, this one has 2. And so when a mite approaches the fly, the way that they respond to the fly is they have these tarsal reflexes. They kick with their legs, and they flick their wings, or they engage in short bursts of flight. And that's their main defense mechanism. Once the mites latch on with their little chelicerae, they have no chance. Like you watch them and they try to groom it off, but it's pretty much hopeless. So this defense behavior is heritable, which we confirmed by doing artificial selection for increased resistance in certain selected lines of flies. The mites also affect host flight endurance, which is not surprising. It's like carrying a small backpack on you and trying to fly great distances. And the flies, they have to fly great distances because the rotting cactus have a very patchy distribution. Not every cactus is good. It has to be at a specific stage of rot for the flies to utilize it. And so in this set of experiments done by he was actually a 299 student. He basically attached a single mite to the fly, and then he tethered it and basically measured how long they could hover without stopping. And you can see the black dot, which represents the infected fly, had a significantly shorter flight duration than an uninfected fly. So we wanted to know whether this was just, wing bearing, just mites interfering with aerodynamics of flight, or if there were other sort of internal physiological effects. So what he did was he let the mites feed for 24 hours and then removed the mites and then immediately tethered them and found that the effect remained the same. So this suggests that it's not just the physical burden of having mites, but that there are physiological costs of infection, and that this is likely to have long-term effects on host dispersal, which could in turn influence gene flow because the flies clearly need to disperse to find new mating habitats. It could influence the population structure at the metapopulation level, and if the populations are segregated enough, that could lead to local adaptations at the metapopulation level. So one of the questions that my students wanted to address is, what are the energetic costs of infection? Now, parasites are known to reduce fitness through these factors, but what about what happens when the host is just exposed to parasites? It's well accepted that infection is harmful, but what about just living in an infectious world, sans infection? So there are costs that are associated with maintaining and activating an immune system, whether it's a behavioral response or immunological. And then there are, of course, costs to tissue repair that could exceed the duration of infection. So one of the first hypotheses we wanted to test is, are there metabolic costs associated with infection that occur prior to actual infection occurring? So in this case with the mites during the pre-attachment period, the period where the flies are exposed to mites but have yet to become infected. So the way we measure metabolic costs is by measuring the metabolic rate, which is estimated by measuring the production of CO2. And we use a system called a small animal flow-through respirometry system. And in its simplest form, you basically put the animal, and in the case of a treatment group, an infected animal with a mite, into a chamber and then you pump air into it, and then you measure how much comes out the other end, and then you take the difference, and that gives you the volume of CO2 that the animal is producing. And you simply multiply that by the rate at which the air is flowing to give you some unit. So you are basically measuring the change in CO2 in the air before exposure, before it enters the chamber, and then you measure the air after the animal has respired. And so the change in CO2 is pretty much what you're measuring. Of course, you have a baseline that serves as a control to account for drift. And then you plug it into this really lovely formula to calculate metabolic rate. So the first experiment looks at how the metabolic rate of flies is influenced by a range of mite infections, so looking for an intensity-dependent effect. So you would expect that the metabolic rate would increase with mite load. So as mite load increases, it imposes greater physiological stress on the fly. And so you basically measure CO2 production during the infection. And I know some of you are going to ask if the mites respire, and of course they do. So, but we put, depending on the number of mites on the fly, we put the same number of mites in the baseline or the blank chamber to account for any contribution of mite respiration. The other experiment that we thought was important to do was to do sort of a before and after experiment. And this is because previous studies done by undergrads show that, interestingly, this is a separate story, but mites actually prefer flies that have a higher metabolic rate. And that may tie in with the metabolic theory of ecology hypothesis, but I won't get into that right now. So one could argue that, oh, maybe the metabolic rate of heavily infected flies, maybe those flies were already exhibiting high metabolic rate, and it's not necessarily a causal effect. So in order to confirm the causal effect, we measure the metabolic rate, the MR of flies before and then after mites were attached. So these results clearly show that there's a linear increase in the metabolic rate or the average CO2 production as the intensity of infection rises. These are the results from the before and after experiment. So they show that, so this value is basically the mean change in CO2 level before and after infection. And you can see that at moderate or low levels of infection and at high levels, the change is significant, and whereas flies that were uninfected, they were exposed, but they were uninfected, and these were unexposed. So in both cases, there were no differences in metabolic rates before and after exposure to the mites. So this suggests that infection increases host respiration in an intensity-dependent manner, and that there are energetic costs associated with infection. So one sort of question that remains is, what exactly is the physiological mechanism, and how does that translate into fitness consequences for the host? So that basically begs the question of what happens when there's no mite on the fly, but the fly is exposed to infection. So we know that infection, obviously, not surprisingly, is energetically costly, but what happens if you're just surrounded by mites and you have yet to be infected? So this was a really clever experimental design that Colin came up with. So he had 3 treatments. He had unexposed mites, so mites that were put into this modified Eppendorf tube. So in one case, there was just the fly by itself. In another case, he had flies that were exposed to mites, but there was no contact between the mites and the fly. So he created, basically using the lid, he created a mesh screen where he put in 2 mites, and then he closed the cap. So there's a mesh on both ends allowing for airflow, so you can put it in the respirometer and measure the change in CO2. And then the blank contained the relevant number of mites depending on the treatment. And then in the third treatment, he left this empty, but he put the mites directly into the chamber where they could come into contact with the susceptible host. So you're measuring the flow, the change in CO2 through the respirometry chamber. And what he found was really cool. He found that, so you have unexposed flies, which have never seen a mite in their life. So this is their rate of CO2 production. And then you have flies that were exposed, but the mites were in a cage. And you can see that they have an elevated metabolic rate, and even more so when they're in direct contact with the fly. So these flies are not even infected yet. They're just freaking out because they hear, smell, or see a mite. So there are metabolic costs associated with infection and with just the exposure to parasites. We don't know the exact mechanism. It may be because the flies are increasing their activity level. They may have physiological changes associated with stress response or some other compensatory physiological response. So it appears that these costs of just avoiding parasites, they may be analogous to what is called the non-consumptive effects that predators have on prey. So some of you probably are aware that when prey are able to detect the presence of a predator, they have increased vigilance, which may take time away from other activities. They exhibit habitat avoidance, and they change where they forage, for example, and they may have implement certain evasive maneuvers, which are obviously, all of these may be energetically costly to them. So we started to explore some of the potential explanations for this elevated level of CO2 production. So this experiment first shows proving some assumptions, which is that indeed the presence here, the mites are exposed, or the flies are exposed to mites, but infection hasn't ensued. So the way that the flies respond is that they groom a lot more, and they start walking around a lot more. Probably because they're trying to evade the mite, they're approaching mites, in comparison to flies that are left unharassed. Now the next thing was to measure the metabolic rate of the grooming behavior, but we had to disentangle the energetic cost of grooming with the energetic cost of being infected. So to do that, we dusted the flies with volcanic ash. So it's a fairly harmless irritant that makes them groom. So just to confirm, indeed, they groom more when they're covered in ash. And then these flies were placed into the respirometry chamber and we measured their CO2 production. And indeed, grooming and having irritants on you is energetically costly. So this may explain why we're seeing a higher rate of metabolism in flies that are exposed to mites, because they're busy grooming and avoiding infection. And of course, this takes energy away from other things, and ultimately that could perturb the way energy is allocated to other resources. But how does that translate into fitness? So, this is a new data actually. We haven't published this yet. So it takes energy away from other activities. So we looked at some of the more energetically demanding activities, such as flight performance. And so in this experiment, the flies were encouraged to groom by coating them with the same irritant, and nearly all of them spent all their time grooming the irritant off, and that took them about 30 minutes to get everything off. And then we tethered them and measured the flight endurance, and you can see that grooming is so energetically costly that it makes them have lower flight performance than if they just sat on resting. So although that's intuitively not surprising, the implications of this is that when you live in an infectious world, it may have implications for dispersal capacity. The other experiment has to do with how does current defenses affect your ability to fight off future infections. So in this experiment, same thing, flies were forced to groom, and then they were exposed to hungry mites. And flies that were forced to groom acquired many more mites on average than flies that were allowed to rest. So it appears that your current investment in defense can negatively affect your future susceptibility to infection. So all of this is to say that there are non-consumptive effects of parasitism, and that these indirect effects can ultimately affect host fitness even if you don't get infected. So I already sort of alluded to this, that this is analogous to the non-consumptive effects that are well documented in predator-prey interactions, where the prey suffer reduced fitness even though they're not actually predated upon. So some of these costs associated with just exposure include habitat avoidance, which may include changes in resource use. It includes induced behavioral defenses, perhaps maintaining immunity. That might apply to other systems. We don't really know if flies respond to mites on an immunological level. That has yet to be investigated. And then, of course, there may be stress responses that ultimately are deleterious to a host fitness. So all this talk about fitness, are there any fitness losses associated with these non-infective, non-consumptive costs? So again, really clever experiment that Colin, he has this knack of doing the experiment, and then when it works, he tells me about it. So luckily there haven't been any major problems. So I was really impressed. I can't take credit for this. So, well, we sort of designed this together, but he came up with the idea of sequestering mites in a cage and exposing them to flies for the entirety of their life. So we use these agar vials where we place female flies, and then if you're familiar with the vials, you know that they come with a foam plug. So we excavated the plug, put the mites in, and closed it off with a mesh. And then the control, same thing, but no mites. And then so in this experiment, he didn't include any males, but then he did another experiment to look at fecundity, so he gave the females some mates. We changed the media just because the flies don't like to lay eggs on agar. They like to lay their eggs on things that remind them of home. So in the longevity experiment, they were monitored until death, and they were transferred to fresh vials every week, and at that time, there were new mites put in, so we didn't have to worry about it. They lived in there for about 5 to 7 days. So we always had live, fresh mites in these cages. And then in the fecundity experiment, he replaced them. He actually gave them 2 males so that the females would not be sperm limited. And then they were allowed to mate, and then every 2 weeks he would give them new males to mate with. And these are the results that he found. So in this slide, you can see that the longevity or days alive shown here is significantly different for flies that had to live in the company of mites compared to flies that were unexposed to mites. In this graph here, it shows the survivorship curve of flies that had to live among mites, and you can see their survivorship drops dramatically at a much faster rate over time than those flies that had no mites in their vials. And ultimately, that translated into lower fecundity. There was a strong fecundity effect, but longevity was an important cofactor, which is not surprising. The longer you live, the more eggs you lay. And as you can see here, the flies that were exposed to mites had a significantly lower egg count over their lifetime than unexposed flies. So these evidence strongly suggest that the mere exposure to infection can have significant fitness effects on the host. So living in an infectious world can carry its own costs, even if you're not actually infected. And for the most part, people doing research in parasite ecology and disease ecology has, not all, but have largely ignored these indirect costs. So we may be, in fact, underestimating the selection pressure that parasites pose on their hosts. The next question that one of the undergrads is tackling is, how do flies detect or sense the mites if they are not, as in the previous case, they're not physically coming into contact with the mites, but somehow they can smell them, chemical signals, vibrations, visual cues. We're not exactly sure, but we're trying to figure that out. One last bit of data that was also, this was done by a summer student. She was very determined. This was not an easy experiment because it took her a long time to work out the experimental design. But what she was interested in is how parasite exposure affects their reproduction as a potential mechanism for understanding the reduction in fecundity. So she wanted to look at whether female flies prefer laying eggs in substrates that had mites or no mites. So she had a large petri dish, a large arena, within which she released 5 female flies and 3 male flies for them to mate with. And then on either side, she placed a smaller dish with the appropriate ovipositioning media. So a little bit of potato flakes, fly media, and a blob of cactus. And they seem to strongly prefer the cactus. But most importantly, she put mites in one side and no mites in the other side in these cages, which are essentially modified micropipette tips. So we don't use Eppendorf tubes and pipette tips the way most people use them. So we go through a ton of pipette tips, mainly because they're perfect mite cages. So that took her a bit of time because it turns out that, so then she had to put a mesh, adhere mesh on either end of that pipette tip. But the problem is that the glue that she was using is toxic to the mites. So we had to create sort of a fancy way of attaching mesh without glue, and which we did finally figure out. And so she then let the flies lay their eggs, and as you can already see, the average number of eggs laid on these dishes was much higher on the side with no mites than on the side that contained mites. So again, the mites are not physically in contact with the fly. The flies are simply detecting the presence of mites, just as prey can detect the presence of predators without actually having to come into contact with them. So it appears that the females preferentially lay eggs in areas that are parasite risk-free. Now, that might be because she's avoiding infection herself, or she may be planning ahead for her offspring. We don't really know why, and that would require further investigation. So the next experiment that she's doing is looking at how strong this bias is when the habitat quality varies. So not all habitats are made equal. In the experiment, both of these habitats, aside from the presence of mites, were identical. But if you give them an inferior habitat that is mite-free, would they choose that over a superior habitat that has mites on it? So sort of these context-dependent decisions that the mites have to make. So just to wrap things up, just to reiterate some of the points, which is that exposure alone is sufficient to generate metabolic costs, which ultimately can have fitness consequences. And this is in line with this concept called the ecology of fear, where there are indirect costs, typically related to predation, but these indirect costs can also arise in the case of parasitism. And the level of defense that a host engages in may also be context dependent. So if they live in an area with high parasite risk, or if the resource abundance varies, then that may affect the level of investment or habitat avoidance that the host engages in. And then related to that is the concept of resource allocation or risk allocation. In other words, if the host can detect and anticipate temporal fluctuations in risk, then can they modulate their responses accordingly to maximize fitness. And then, of course, this is going to have implications for foraging, dispersal, and mating decisions. So my last slide, conclusion, what we know so far and future directions. So it's clear that ectoparasites can exert a fitness cost on their host, and that these are likely mediated through perturbations to the host's energy budget. We know that parasites can reduce flight endurance, and it might be interesting to look at how that affects host dispersal, especially in the field. And given that mites can only disperse on flies, this is ultimately going to affect mite dispersal as well. So there may be sort of a threshold where we know that mites, as a side point, mites also prefer flies that are already infected. But at the same token, heavily infected flies don't disperse very far. So it's kind of a contradiction, and there's likely a threshold which we haven't tested because it turns out it's really hard to get mites to infect flies when they're in the pipette tip. So generating high levels of infection is a challenge. But there is likely some threshold where the benefits of this high level of aggregation gets outweighed by the costs. So, and we know that given that defenses are costly, they're going to trade off with other activities such as flight, resistance, future resistance, and other components of their life history. And finally, just as a side point that we haven't actually addressed, which is that when parasites influence host behavior, habitat use, that's going to have a knock-on effect in the community. So when hosts change their behavior and change their habitat use, that's going to change how they affect and how they interact with other members of their community, with other competitors, other predators, and so on. So there may be sort of parasite-mediated, trait-mediated indirect effects due to infection. So that's all I have. Any questions? Please don't ask me anything about the respiratory system. As I told Maya, I don't even know where the on switch is.
Lots of time.
Yes?
So when you're doing the CO2, the respirometry measurements, I couldn't see how you would distinguish between the flies using more energy and the mites responding to the presence of the flies, but we have more CO2. I mean, it's possible the mites get very excited and use a lot of energy trying to get at the flies.
Yeah, that's a really good question. So when mites attach to the flies, they're presumably feeding, and that may actually increase their metabolic rate over what you would measure in a mite sitting in a chamber by itself. So we thought about that, and we've actually done some sort of experiments to verify, and it does go up a teeny tiny bit, but it's on a scale that's so small that it doesn't really register in the system.
I'm talking about exposed but not infected case.
Where hosts are exposed.
Exposed.
Oh, so in those experiments,
Still in the chamber, right?
right? So in those experiments, yeah, you're right. So the mites that perceive the presence of a host might respire more because they're excited or whatever. Yeah, so we've done similar sort of little pilot experiments, and again, it's so minor that it doesn't really change, for the resolution that we have, it doesn't really affect the overall output by the fly. Yeah. And in fact, we have to jam in 10 mites to even read it because the levels are so low. Yeah. Yes?
You mentioned that you don't know how the fly senses the presence of mites. Have you tried doing the experiments in pitch blackness to take out vision, or with an overwhelming smell to take out smell?
Yeah, so I'm trying to get a USRA student to do that. So that's a great experiment, just to eliminate each one of the cues sequentially and see which one is playing a more important role. Yeah. Yes, John?
Along the same questions, do you get the same response if you use dead mites versus live mites?
We've considered using dead mites, but since we don't know the precise mechanism for how the flies detect mites, we wanted to just use live mites. But if we were trying to zone in on the mechanism of detection, in other words, if we want to eliminate the possibility that flies are cueing in on the vibrations that mites are producing, we could use dead mites.
Cueing in on the movement,
Yeah.
so that they're hearing them.
Right.
So
Right.
you can eliminate that
Yep.
as a possibility.
Yep. So that could be one of the factors that we.
So when it comes down to it, what do you think is the cue?
So I think it's chemical cues. Yeah. Simply because flies, so much of their life is chemically mediated, and the mesh that we use is kind of opaque, so it'd be surprising if they can see through it. So most likely it's chemical cues.
Is there anything known about which chemicals might be involved?
No. Yes?
You said that the mites prefer lightly infected flies.
Mhm.
And I wonder, why would that be, and how general do you think that is for parasites?
Well, that really only pertains to parasites that can actively seek out their hosts. So there are a number of parasites that are passively ingested. So in those cases, the parasite doesn't really have a preference. They can't really exhibit host preference. As far as why, we don't really know, and that's part of Colin's PhD dissertation. He's trying to measure the fitness effects of the fitness consequences of these preferences. From a purely conceptual perspective, it could be that, so the mites, when they attach to the flies, they're already gravid, or most of them are. They're already mated, they have eggs, so when they arrive at their new habitat, they're ready to lay their eggs. So they don't need to find a mate necessarily when they get there, but they probably want mates for their offspring. So females, they're haplodiploid, so they can mate with their sons, so they can have sons even if they're not mated. So if they arrive as virgins, they produce sons, and then they can mate with their sons. But that's probably less ideal than mating with another conspecific. So it could be an adaptation to a certain extent, but there should be a tipping point, you would expect, where too many mites on one fly results in competition, reduced dispersal capacity. Could be any one of those.
So maybe some sort of general outbreeding potential with sort of this optimum infection rate.
Right.
for the offspring,
Yeah.
I think.
Yeah. Yes. Yes.
Would it be possible to take 100 mites, mush them up and make odor mite, and then dose the flies with the correct volume for 1, 2, 3
Yep.
mites? And you still don't know whether you get any results or not, but it might be fun to try.
Yep. I've tried pitching that to an undergrad. So we do have some really small pestle and mortars, so just the fun of mashing them. Although it won't be easy, would it, Heather, because they're
I
quite.
think you just want to wash them
Yeah.
in an
Yeah.
organic solvent rather than mush them.
Yeah, so we've been talking about doing like a wash of some sort. Yeah.
Yeah.
Although mashing them seems equally fun. Yeah. Yes, Stan.
So could all these costs that you've shown be a function of the fact that you haven't given them an alternative option, which is to basically fly away and avoid the mites completely?
Right.
And expend the energy that
Yeah.
way. If you don't let them do that, then clearly they
Yeah.
have to do all these behaviors to try and resist being infected.
Yeah.
And
So.
that's going to up your expected or
Yeah.
your costs as you view them
Mhm.
for these non-consumptive effects.
Yeah, that's a really good question and comment, which is that we haven't given them any other option. So the amount of costs we're detecting are probably overestimates. But ultimately it translates into another cost, which is the cost of leaving the habitat that would otherwise be ideal for reproducing on. So if they arrive at a site that has heavy mite load,
It's not ideal to reproduce on. They should get that
Right.
a lot of dodge, as you
right,
said.
but yet they do. We see them in the field. They still court females in those sites. They still lay eggs. So there's some complex trade-off that they're making, right? And we have to, I mean, that would be a really neat experiment to offer them alternatives, like in the ovipositioning experiment. You know, if we give them an optimal habitat to oviposit on, but with a certain number of mites that varies, and see how that affects their decision making. Yeah. Yes, Maya.
You could kind of test what Stan's suggesting by just exposing them and then putting them on the tether and seeing if exposure increases dispersal, right?
Yes. So we kind of did that with the grooming experiment, but to do it more realistically with, oh, I see what you're saying. So just
for
have
the
them,
flight
yeah,
behavior.
and see if they disperse further.
Yeah, so
Okay,
give them a
yeah.
pre-exposure
Right,
to the mites
okay, that's-
and then
yeah.
put them on the tether.
Okay, more undergrad projects. Yes, Rich.
So I love the image of a fruit fly on a leash. And I have a question about the leash. Is it a really ultra-fine filament?
Yeah, so initially we were using,
Fairly stiff, so the flies.
it was fairly stiff. Initially we were using fishing line from fly fishing. So you can imagine me in a sporting goods shop, asking them for the finest fishing line possible because I look like a fly fisherman. But ultimately we switched to another material, tungsten wire, which is more stiff, lighter, and you can order it in like 0.1 millimeters or something ridiculously thin. And originally we were using super glue, but now we use UV cured glue. So we've refined our technique.
So the reason for asking about the tether was I was thinking about you show an added or shorter duration flights after they're parasitized. But if they're on a tether, the tether is suspending the weight of the fly and the mite. So there isn't like the added loading of the extra weight of carrying the mite. So I'm wondering why do the flies fly less if they're not carrying that extra weight?
Well, when the mite is on them, they're probably feeding on them. So it might ultimately come down to the physiological cost of having someone suck on your blood while in flight. Yes, Rich.
Following up a bit on Stan's question, I guess, thinking about attempting to scale up with these putative costs, etc. If we were to look at that picture and imagine all the flies that are in that picture, they're hard to see, but what proportion of Drosophila at any point in time would be infected by these guys in that picture on that scale?
So in the wild, in nature, it varies depending on the age of the rot. There's a direct relationship. As the rot increases, you get higher prevalence, but then you start to see these high levels of aggregation. So the necrotic cactus, they eventually dry out. That's inevitable. So if you go in the field and you go to a really dry rot, you'll collect flies with tons of mites on them. It's fairly common. But if you go to a rot that's relatively fresh, they may be mostly uninfected or they may have 1 or 2 mites on them. on the flies. So it's not uncommon to see that level of aggregation, but again, it's directly related to the age of the rot.
Sure, but if you're going to make an argument that these things may have an influence on, say, gene flow, population level, in that picture, if you said out of those 500 cacti, 4 of them at any point in time are heavily rotted, one would question the ability to scale up these things. That's what I'm trying to get at.
I'm not really sure what you're asking.
It's a very rare event, I guess, on an ecological scale, i.e. the heavy rot. If the proportion of heavily rotted cacti is very low in any given...
Yes, but in terms of exposure, I don't know the degree of exposure in the field. When I sample flies, I just know how many mites are on them. But when a rot, at any stage of the rot, there may be lots of mites running around. So the flies are potentially exposed to lots of mites, not necessarily at the tail end of the rot. So the pressure imposed by, so when the flies arrive, they bring, to their own detriment, they bring the mites with them. And then the mites get off and they reproduce like crazy in the rot. They're haplodiploid, they have short generation, one generation's like 5 days. And so mite populations can expand rapidly within the fly's lifetime. And so they're readily, they bring the mites with them, so already they're in an infectious world potentially.
Right.
Yeah. Yes, Andy?
So when you capture the flies in the field, do they all have mites on them?
Not all, and the percent depends on the rot. So when I arrive at a dry, really, like the first picture you saw where it's fallen over and it looks like a skeleton of cacti, those flies were like 60% of them were infected. But if it's a fresh rot, I'd be lucky if I see 10, 20%. So it really varies. Yeah. Yes, Heather?
So you've showed that for whatever reason, the flies are able to detect the presence of caged mites and it makes them anxious. Have you considered trying to present them with mesostigmata that are not going to be attacking them, like with phytosaids, to see if it's just general anxiety about stuff?
Yes.
There's stuff moving.
Yeah.
Are they really worried about Macrocules subvadius, or are they just worried about the squeaky noise and the smell?
Yeah, that's a really excellent point.
You can buy phytosaids.
Okay.
Right? They're biocontrol agents, and Andy probably knows who rears them for greenhouse control, and they're mesostigmata.
Yeah.
But they would not feed on fly larvae, and they would feed on flies.
Yeah. Yeah, so we've talked about introducing other natural enemies and conspecifics even, and non-parasitic mites to see if we can get the same response. Yeah. Yes, Andy?
So the nematodes insect pathogenic or mite pathogenic?
No, the nematodes are really cool. They're phoretic, and they enter the flies during when the flies are forming their pupae, and they enter like this, I don't know what you call it, maybe the Drosophila people know, but they have this little pocket in their head that inverts. So, what? Okay. Yeah, so the nematodes go in there, and then when the flies die, they come out.
Why don't you try an fly pathogenic nematode?
To test what question?
Well, see what role they could play. They wouldn't cause 100% mortality.
Oh, okay.
So the nematodes always get there near the fly.
Yes. Yeah. 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 It would be interesting to see how the nematodes interact, but we don't know how to culture the nematodes, so we're not quite there yet. Yes, John?
Sorry, couple more questions.
Sure.
So what's the life cycle time for the mite versus the fly?
So the mite, a new generation every 5 days. So it's really rapid.
And the fly?
The fly, for the Nigrosporacula, about 20, 21 days.
Okay, so you
Yeah.
have essentially 4 generations in.
Yeah, yeah.
Another question. When you deal with the flies, you have to put them under CO2 to
Mhm.
move them.
Yeah.
So what happens with the mites that are on the fly?
Oh,
Do they
so
fall
they
off?
fall off.
So they go to sleep as well?
Yeah, so we always, we're careful to set up an experiment where the mite is the last step. So if we need to CO2 flies for whatever reason to tether them, we do that first, and then we let the flies recover before we introduce the mites. So we never, for various experiments, we know that the mites fall off. So in some cases we use that to our advantage, but we always plan things such that we don't lose mites prematurely.
Yeah, I mean, fruit flies can also be adversely affected by CO2.
They can, and
Do the mites are equally adversely affected, or do you know?
I do not know. And we know that metabolic rate is going to be affected by CO2 exposure, and we considered using like cold as a form of anesthetic, and that also has an effect on their CO2. So, but we're not interested in the absolute level of their metabolic rate. We want to compare them to control flies to see the effects of parasitism. So for our work, we kind of don't worry about that.
Sorry, one more quick question.
Yeah.
How inbred are your strains?
Of flies?
Yes.
So we breed them in mass culture, and they've been in the lab for 2 years. And we maintain a population of 100, I can't do the math, 100,
One pair,
200.
one pair to isogenize or try and isogenize?
No, they're all just all one panmictic population, yeah, of 200 plus flies, yeah. Yes?
Apropos of the question here about CO2 effects, do you treat your controls and treatments the same way with CO2?
Yes, so we do everything identical, yeah. Yeah. Yes, Rich?
Just to follow up on that, what was in my mind is that experiment when you add mites to the portable guys. Do you have a procedural control where you actually handle the flies and pretend like you're attaching mites?
Well, we don't actually attach the mites ourselves. We just put them into these really tight micropipette tips where the mites can't exhibit any form of resistance. So we eliminate that factor. So that facilitates infection. And then we do the same thing to the controls. We shove them into the pipette tips. They're like little straitjackets. We shove them in, yeah. Yes, Stan?
Is this all run through animal care programs?
They're not real animals, right? We have a couple of fly moorings in our lab, which I'm sure wouldn't pass animal protocol. All right, thank you, everyone.
Before you all run, don't forget there are drinks and food nibbly still, and for you, a symbol of the department's undying appreciation for your gift this afternoon. Yeah, treasure that. All right. And we will